let's talk about the seed. And at the beginning, let us talk about the components of the seed. Each of the seed consists of three components. What are those? Just like what you had seen in the previous case of the embryo, the first component is going to be the embryonal axis, second the cotyledons, and the third a layer, that is a seed coat that covers both of these. Now when we talk about the seed coat, as you have learned in class 11, it is the intestine segment, that is two layers that form the seed coat. Now we all know that the ovule forms the seed, and all the components inside may or may not be present in the final or mature form of seed. Now the question is, what about endosperm? Now the endosperm might be completely used up, or it may be present to some amount, and depending on that we are having two categories of the seed. One is called the non-albuminous or the S-albuminous seeds, and here the seeds typically do not have any form of endosperm, that is, there is no residual endosperm in the seeds, and we can see that in case of the groundnuts or the pea. You have seen the seeds, I guess. Now the second category of the seed is the albuminous seeds, and in this there is some amount of residual endosperm present even in the matured form of seeds, and that is typically seen in case of the wheat, maize, barley, castors. Again, these examples are directly from the NCERT. You have to remember because they can ask direct questions from here in the examinations. Now you also have new cellus in the beginning when the development of the this embryo sac had started. New cellus, if you remember, and in some cases of the seeds there is also some amount of new cellus present in the matured form of the seed, that is the persistent in the seed, and that is called the perisperm. Now what is perisperm? It is a persistent form of new cellus present in some of the seeds of some plant species. For example, in case of black pepper and beet, you have the perisperm. Now do not confuse this with the pericarp. Pericarp is something different, that is the wall of the fruit. Perisperm is a persistent nucellus, and nucellus is not same with the endosperm. Nucellus provides nourishment to the developing embryo sac, right? It gives rise to the, in the beginning, gave rise to the MMC, megaspore mother cell, if you remember. Endosperm is going to form much later. Now, what are the fate of different components that we had seen till now? The integuments that we had, integuments typically gets converted into a hard covering, and it is the seed coat, right? The ovules develop in the seed, the ovary develops in the fruit, and the walls of the ovary that develops in the pericarp or the wall of the fruit. As I was saying, perisperm and pericarp do not diffuse, sound similar, but they are pretty different. Okay, so these are the concepts that you have to understand, basic concepts you have to understand regarding the seed, how it is forming. Integuments going to form the seed coat, walls of the ovary going to form the pericarp or the walls of the fruit. Remember this. Now again, here these are the images from the texts. You can see some of the structural seeds on the top, right? A dicot, a monocot. You can see different structure, that is the seed coat, right? Endosperm, cotyledons, and how the hypocotyle and apical structures are organized. And again, this is the structure of the monocot seed from class 11, so nothing new for you. It is all class 11 concepts. Now what happens when the seed is matured? Remember, the seed, once it is matured, has to be dispersed by process of seed dispersal. Different plants have adapted to different seed dispersal mechanism, and each of the seed dispersal mechanism has only one aim or one goal, to allow the seeds to move or to be transported to a variety of places to a long distance, so that the seeds have a very good chance of germinating and surviving. Now what happens in the seed dormancy? When it is in the nature, when it is dispersed, it typically enters a stage of seed dormancy. Now it becomes dry, that is the water content is almost reduced, and the seed becomes relatively dry. The general metabolic activities on the cells of the embryo, it also slows down. Now till what time does it occur? It occurs in the dormant stage till a favorable condition is going to return, and till the time the seed is going to ready for germination. And during germination, the seed, dormant seed, it takes in the water through the micropyle, through the small minute pore in the seed. It swells up, the oxygen is going to be taken in, and the metabolic activities become much rapid, back to the normal, and it starts to develop, it starts to germinate. Now, in some cases, the fruit that is formed are not true fruit. So we have to understand what is true fruit first. True fruit means, we all know, in the previous slide we saw that the fruits are typically developed from the ovary. Now, any structure, any fruit that is developed from structures other than the ovary, for example, the thalamus or any other structure, that is called a false fruit. They look like fruit, but they are not the actual fruit. The actual fruit is much reduced. As I said, fruit is a structure formed from the ovary, right? And if anything that is going to form looks like fruit but is not developed from the ovary, structure that is other than ovary given it to the fruit, they are called the false fruit. As you can see here, in case of apple, the entire fruit that is consumable typically, they are actually the thalamus. It's an example of a false fruit. Similarly, even in case of strawberry, the fruit is not a true fruit, it is a false fruit. So that's the difference between a true fruit and a false fruit. Now, in some cases, you have seen this fruits that we get in the market. For example, grapes are typically nowadays, they do not have seeds, or seedless fruits, seedless grapes, right? And you can see in this image there is a seedless watermelon. Now, how do we get these seedless fruits? Now, it is a naturally occurring process. Again, this also can be induced artificially, and the process is called parthenocarpy. Now, what is parthenocarpy? It is the development of the fruits without fertilization. So, development of the fruits. So we know that if only fertilization will occur, then only we'll have the fertilized egg and ovule will be there, that will give rise to the seed. But if you do not have fertilization, then there is no chance that you'll going to get the typical seed as you get in a normal condition. So what is the benefit? It benefits is it produces seedless fruits, and that is typically of importance, especially when it comes to the consumption by humans. Now, as I said, artificially we can induce this process with the application of certain growth hormones. And some of the examples of naturally occurring parthenocarpy is pineapple, banana, cucumber, grapes, orange, right? These are some of the naturally occurring parthenocarpy. Now, do not confuse, do not think that the process of parthenocarpy and the process of parthenogenesis are the one and the same. No, they're related, but they're not the same. We will talk about parthenogenesis in the upcoming chapters. But in case of plants, development of the fruits without fertilization, remember, it is called the parthenocarpy. Now, when it comes to seed and the advantage, there are few points that I'll just tell you. I have taken this entire thing from the CBSE directly, NCERT book directly. Now, pollination and fertilization are independent of the water in the angiospermic plants, and that's why the seed formation is more dependable in case of these plants. The seeds have better adaptive strategies for the dispersal, as I was talking about, to new habitats so that they can germinate and they can increase their number. As they are having rich food material reserved in the form of, let's say, cotyledons, they can nourish the young seedlings in a much better manner. The hard seed coat, it provides a protection to the embryo. Okay, it is going to provide protection to the young, basically delicate embryo inside it. As it is going to be a result of the sexual mode of reproduction, it is going to produce new genetic combination, and that is going to lead into variations. Now, what about the seed viability? Yes, the seed have been dispersed as we are talking about, but what about seed viability? How much time can it survive? Because if it is dead within minutes of release, then there is no point. Just like pollen viability, there is a seed viability also, and again it differs in a diverse manner from one species to another. We'll just talk about two of the oldest. One of the oldest recorded viable seed is the lupin. It has been excavated from the Arctic tundra, and it had a dormancy. It germinated over after 10,000 years, right? It had a 10,000 years of dormancy. That means it was basically viable after dispersal for almost a long period of time of 10,000 years. One of the recent one that we have found is a date palm, Again, that also is basically an old, I mean, they can survive for almost 2000 years, and the seed is going to be viable for almost 2000 years. That is Phoenix dactylifer, that is a scientific name. So again, as I said, the seed viability differs to a greater extent. Some of them can survive for thousands of years, as you can see in this particular case. Now we are coming to the end of the entire process. We said about the process of formation of megaspore, microspore. We said about pollination. We said about fertilization, double fertilization, right? And we also talk about the formation of the zygote in the embryogenesis, and we said the formation of the seed, right? And that completes the entire process of reproduction in flowering plants. Now, why did I bring up this image? I just wanted to show you the process, how it occurs in relation to the life cycle in the plants, as you have learned in the class 11th. In class 11th, you have learned about three different life cycles in plants. What are those? One is called the haplontic, another is called diplontic, and another is called haplodiplontic. Now, as you can see here, the plant at most of the time it spend in the form of a sporophytic diploid condition, a two-end condition, and for a short period only it becomes the haploid. Okay, they form the gametophyte generation. And again, if you remember, it is a diplontic life cycle in case of angiospermic plants. Again, it's a class 11th concept. So only when the megasporangia and microsporangia, the structures undergo meiosis, you start getting the haploid structure, and only then you have the gametophytic stage, right? And till what? Till the double fertilization. Once the double fertilization is over, then the gametophytic generation is over. It is short-lived generation, and it is kind of dependent on that of the sporophytic generation. So that is the entire process of the flowering plant life cycle, right? And remember, both gametophytes are highly reduced to a few cells only. Okay, let's complete the process. Now we have another concepts that are slightly related to this. Let us have a look of the apomixis and polyembryony, and with that we'll be completing this particular concepts on reproduction. Apomixis is a form of asexual reproduction that mimics sexual reproduction. Now, why do we say that? Because in this process we have the seed formation, but it is produced without fertilization. Now, parthenocarpy, if you remember, that is the process of formation of fruit without fertilization, but here we are having the seed formation without fertilization. Typically, what should happen, we have learned that seed is formed only after sexual reproduction, but by this special process, some species of the Asteraceae and some of the grass species can produce the seeds, but without fertilization. So how does it do it? One of that method is, in some cases, the diploid egg cell is produced. Without meiosis, if the egg cell is produced, it will be in 2N form, and this 2N diploid egg cell directly develop into embryo without fertilization, and as it develops, it gives rise to the seed also. The second method is the involvement of the nucellary cells. Now we all know that outside the embryo sac, the nucellus is the nutritive tissue, and in some cases of citrus like lemon and mango, they start dividing and they protrude into the embryo sac, and then they develop into the embryo. Now in such cases, typically we see more than one embryo inside a single ovule. Now till now we have learned that a single embryo is present inside a single ovule, but in these cases you have more than one embryo in a single ovule, and this process is called this condition is called polyembryony. Poly means many, embryony, many embryo, more than one embryo per ovule. This is not common. Again, typically we get one embryo per one ovule, right? So this is the case of polyembryony. Now why are these important? Why this process of apomixis important? Let us understand that. So we all know hybrid seeds are very important. It has helped the farmers to produce a lot more crops in the same given lands, and typically the hybrids are produced in such a way that the farmers have to buy the seeds every year. There is a reason to that. Now the hybrid seeds basically need to be produced every year. Why? Because the seeds that we obtain from hybrid plants, when we grow a hybrid plant and we obtain the seeds and then I try to grow it again, then typically I don't get the hybrid traits. Okay, the hybrid traits are lost because when we grow these seeds, the genes, the traits, the good traits, the hybrid traits, they actually segregate because they undergo sexual reproduction. They segregate and they lose the hybrid traits. So the yield from the hybrid seeds directly will be when you compare the yield from a hybrid seed and when we compare the yield from a seed that has been grown from these hybrid plants, then the yield is much lesser when we grow it from the product of the hybrid plant, the seeds of the hybrid plants. And that is why every time the farmer, they have to buy the hybrid seeds, and that basically the hybrid seeds are not cheap, they are basically pretty costly, and that's why the farmers need to spend a lot of money to just buy these hybrid seeds. And you know the condition in our country, if we can provide hybrid seeds to the farmers at a lower rate, it will be much better for our country. Now there's a cost factor involved. So these are the problems with the hybrid seeds. Now what is the solution? Apomixis might be a optimum solution to this. How? If we can produce apomictic hybrid seeds, right? Why? See, if we can produce these, as there is no fertilization in the process of apomixis, there is no chance of this segregation of the genes. So there is no chance that the hybrid traits are going to be segregated out, and we are not going to, and because of that we are not going to lose out the hybrid characters. And that is why when we are going to create a apomictic seed, that apomictic seed can be used again and again with the hybrid characters.